Mounds of hydrated silica on the sides of Patera caldera suggest the presence of past hydrothermal activity, which could have provided a habitable environment for past extremophiles, even when the rest of Mars had already become dry and cold. The Patera caldera is part of the Syrtis Major volcanic complex, which formed approximately 3.7 billion years ago during the early Hesperian period. The Syrtis Major complex is thought to have been active for approximately 100 million years, with hydrothermal systems active throughout this entire time. This would have generated multiple surface springs, each of which could have supported life. Martian water was often acidic, especially from rocks of this age range. It is likely then that the water in these hydrothermal vents was also very acidic. Ancient Martian environments were often high in sulfuric acid. Silica hot springs are a particularly good place to look for evidence of ancient microorganisms. Silica sinter can preserve body fossils, biofilm, and microbial mats, biological sedimentary structures such as stromatolites, chemical fossils, and biogenic isotope ratios.
Norris Geyser Basin is the oldest and hottest geyser basin in Yellowstone. Most of the water in the basin is acidic. This is rare for the park, as most of the geyser basins there are alkaline. Furthermore, the water in the Norris Geyser Basin has been shown to have high levels of dissolved sulfate, indicating the presence of sulfuric acid. On Earth, microbial mats often form stromatolite-like layered formations. It is likely that these would have survived on Mars. The oldest known silica stromatolites from the Dresser Formation in Western Australia are around 3.5 billion years old. Especially if cells were completely entombed within the silica sinter, there is a possibility that chemical biosignatures may have been preserved. There is a danger, however, that UV radiation could have destroyed some of this, and experiments will have to be formed with this in mind. Even so, the sheer size of the opaline silica deposits does suggest that microbial fossils and stromatolites could have still been preserved.
This bad boy is Sulfolobus acidocaldarius. This is an aerobic thermoacidophilic archaea with a preferred habitat of 80 degrees Celsius in pH 2 in terrestrial hot springs. It is able to metabolize sulfur from hydrogen sulfides, producing sulfuric acid as a waste product. It is found in acidic hot springs in Yellowstone National Park. Most of the sulfuric acid on Earth is due to bacteria that eat sulfur and convert it. Mars has been identified to have high levels of sulfides, which could indicate the presence of some microbes on the planet. By lacing an isotopic sulfur sample, a solution containing sulfur 35 and oxygen, and allowing a rover to inject the sample into the soil, we can check for the presence of life by observing the evolved gas. Analysis of sulfur isotope ratios can suggest the presence of ancient sulfur metabolizing organisms. The rover would be able to differentiate between the isotope and normal sulfur using a radioactive counter and compare the results with a soil sample from Yellowstone with known microbes, treated under similar conditions to Mars. Because abiotic processes can also result in the presence of skewed isotopic ratios, this technique should be used to confirm the past presence of microbes in samples which have already been identified as probably containing microfossils. If the production rates match, it could be possible for the acidophile to thrive on Mars.
Time-of-flight secondary ion mass spectrometry, or TOF-SIMS, can detect organic molecules in ancient fossils. This is a promising technique, since lipid biomarkers can survive for billions of years in sedimentary rocks. TOF-SIMS can identify organic molecules and correlate their spatial distribution with biogenic morphological features. Furthermore, because the current goal of discovery of life on Mars does not have to exactly identify the type of microbe present, only identify that it was there, we propose that TOF-SIMS could be used to search for remnants of life in the Patera caldera. Depending on if we can get a machine on a Mars rover, this experiment might require sample return. Mars 2020 is capable of sample return, but would have to be targeted to the hydrothermal formations.